Good morning, everyone. Though I know people are still gonna be wandering in for some time, I wanted to go ahead and get started. Good morning, good morning. I wanna make sure that we leave plenty of time. We have the great fortune today of having an esteemed guest, Dr. Josh Gordon, a wonderful friend, really returning home to UCSF after many years. We've been trying to get him here for a while. He was a little busy leading the NIMH, but now he's got a lot of downtime leading the Department of Psychiatry and Behavioral Sciences or is it both? The Department of Psychiatry. I don't want to get it wrong. It's just Department of Psychiatry at Vagelos. The more formal introduction is to tell you that Dr. Gordon is accurately described, I believe, by ChatGPT as a visionary neuroscientist and psychiatrist. He received his MD and PhD degrees in neuroscience from here. Completed a psychiatry residency and research fellowship, the latter research fellowship with Renee Henn at Columbia, prior to serving on the faculty there in the Department of Psychiatry from 2004 to 2016. He moved to the National Institute of Mental Health, where then he served as director. He had an extraordinary tenure as director, championing the integration of neuroscience and clinical practice and advocating for something that I know many in this audience are intensely interested in, which is the move towards precision medicine approaches to tailor treatments to patients based not only the unique genetic and biological, but also their psychological profiles. In addition, again, I think familiar to those of us here at UCSF, or at least gratifying to see NIMH move in this direction, that he was a leader in advancing and addressing the importance of moving forward with enhancing diversity and decreasing disparities in mental health care at a national level. In addition, he had a major initiative in enhancing suicide prevention research. All the while, Dr. Gordon has remained an active researcher. His lab studies genetic models of psychiatric illness from an integrative neuroscience perspective, employing a wide range of modern tools with significant consequences and potential impact in a variety of psychiatric disorders, including psychosis. As I mentioned, he did leave NIMH and return now to Columbia, where he serves as the chair of the Department of Psychiatry, Columbia Vagelos College of Physicians and Surgeons. He's the executive director of the New York State Psychiatric Institute and Psychiatrist-in-Chief at York-Presbyterian Hospital, Columbia University, Irving Medical Center. It is truly a wonderful pleasure to have you here to talk to us today about a broad view of past, present, and future. Thank you, Josh, so much for coming.
Thanks, Matt. Thanks, Matt. Matt and I have been friends for a long time. I think the first time we met, we were nominally trying to recruit you to Columbia for a genetics position early on in your career and much earlier on in my career. And it's been wonderful. When Michael Stryker, who most of you probably know, came to me at SFN in 2015 and tell me I should apply for the NIMH job. The first thought I had was, what about Matt State? And he said, well, he's new to UCSF. He's not going anywhere, so you better apply. It is a pleasure to be back here at UCSF. My first exposure to psychiatry patients was over at Langley Porter. I did a rotation at the VA, but it's really nice to see a beautiful building that, to quote Matt, shows that you can just break the stigma of mental illness by building a beautiful building, which is wonderful. I want to talk to you today about neuroscience and mental health. And I recognize that the main message that I'm going to assert, that neuroscience is important for psychiatry, is not an unfamiliar message, and probably one that all of you in this room accept on faith. But at least one part of the message they're going to impart is one that we implicitly and sometimes explicitly don't take on faith. And that is the notion that neuroscience has already made contributions. So I'm starting with neuroscience past, and then moving on to present and future. I have to say in the disclosures that I'm an advisory board member of NeuroCare, and a shareholder in Integral to neuro psych startups. Nothing I say today is going to have anything to do with the work that I do with those two companies. So the agenda for the talk, I'm going to try to make the case in the first part of the talk by way of introduction that neuroscience has already made contributions to mental health. And I think that's the part that many of us don't necessarily take on faith. So I want to give you some evidence for that, and you can take it or leave it for how important those contributions have been. But I think too often a seminar is started out by saying, we don't know anything about X psychiatric disorder, or neuroscience has not led to any novel treatments in psychiatry. And I think it's important that we take credit for how neuroscience has changed the practice of psychiatry already in order to justify and continue justifying neuroscience investments now and in the future. Then I'll tell you my own neuroscience story from my lab as a neuroscience present message, and then I'll spend the last 10 or 15 minutes or so talking about how I think neuroscience will influence psychiatry in the future. So when we think about neuroscience contributions to mental health, I want to recognize that, or make the case that even though many of our treatments initially were started by serendipity, that there's a neuroscience basis for most of them as well. One good example of that starts with this gentleman in the middle of the picture with the eye patch on one of his eyes. Of course, many of you know that's Julie Axelrod, who was a scientist at the National Institute of Mental Health for decades. And in 1970, he was awarded the Nobel Prize in Physiology or Medicine for research on the chemistry of nerve transmission. And in particular, one aspect of neurotransmission that he studied was how neurotransmitters are broken down or gotten rid of from the synapse, including the process by which the reuptake of neurotransmitters happens into neurons. And it was his work that enabled, after the more or less accidental discovery, and we'll talk about the more in just a moment, of MAOIs and tricyclic antidepressants as the first classes of antidepressants. His work allowed the recognition that those compounds affected the degradation and reuptake of monoamines. And so every single other antidepressant since, which have had reduced side effects, higher safety, better tolerability across the board, if not better efficacy, every single one since arises directly from the neuroscience knowledge that Julie Axelrod and others made that enabled us to build better molecules with more specificity and less side effects. A similar example is true for antipsychotics. Of course, we all know that that antipsychotics, at the very least, they all bind the D2 receptor and antagonize it. The first antipsychotic, chlorpromazine, was more or less, again, we'll get to more in just a moment, was more or less discovered by accident. It was thought to be it was tried out as a sedative, and it turns out that a sedative given to psychotic patients actually reduced their hallucinations. But it was the discovery that chlorpromazine and other early antipsychotics that were chemically derived from it blocked the D2 receptor, and here the recognition that the dose at which they were effective on the x-axis correlated with their inhibitory concentration on the y-axis very, very strongly. That led again to the development of more specific D2 receptor antagonists, with better side effect profiles and greater efficacy actually than chlorpromazine. And so the neuroscience of nerve transmission, to quote Julie Axelrod's Nobel Prize, led to the proliferation of a number of different options for psychiatrists. And if you think about it, every drug we use today for psychosis and for depression originates from that neuroscientific knowledge that allowed us to build beyond those first serendipitous discoveries. A note on the more. In particular, the antidepressants, but to a lesser extent also chlorpromazine, they weren't completely accidental. There was a histaminergic hypothesis, for example, of depression that led people to test the MAOIs and the TCAs. It turned out to be a wrong hypothesis and the wrong neurotransmitter, but it was still a hypothesis-driven set of experiments that led to the development of those drugs, as Steve Hyman has pointed out in a review he wrote about 10 years ago. Now, we classically think of, and we want to imagine, that there's a more principled way of developing treatments for any illness, but of course for mental illnesses, way that might derive directly from basic neuroscience principles. So I just told you that, an accidental, more or less, discovery of an initial drug led to a proliferation of lots of other options through our neuroscience knowledge. But that seems less satisfying to us who are toiling away at basic neuroscience, trying to figure out what circuits and what molecules are involved in symptoms that we might want to treat in our patients. And heretofore, it's been difficult to identify such a classic, identify a mechanism, develop something that targets that mechanism, and treat that disorder story. In 2017, though, the FDA approved brexanolone, and I would argue that it is, if not the first, one of the first psychiatric treatments that derives from basic science knowledge all the way through to clinical hypothesis from that knowledge on to treatment. And it starts with the notion, as shown on this slide, work done, in this case also at the NIMH, led by Steve Paul and others, to examine the mechanisms by which neurosteroids, naturally occurring steroid neurotransmitters in the brain, affect neurotransmission. In this case, they showed, if you look at V1 on the lower left of the graph, that you could take GABA currents and in vitro, and those are the little downward lines here, and enhance them by applying one particular neurosteroid, allopregnanolone, into the bath around your neuron that you were stimulating. And this potentiation of GABA actually is quite similar to the potentiation of GABA currents that you get with benzodiazepines, although it was shown over the years that the neurosteroids actually bind to a different site on the GABA-A receptor. Now, it turns out that through biochemically, it was shown that allopregnanolone derives from progesterone. And it turns out also that these neurosteroids, and in particular allopregnanolone, can be stress protective. So if you stressed animals and you gave them allopregnanolone, it would reduce the subsequent behavioral impacts of the stress. And so that led to the hypothesis that maybe allopregnanolone might play a role in postpartum depression. Why postpartum depression? Because, of course, progesterone levels drop precipitously after pregnancy. And it had been shown actually that in the brain, allopregnanolone levels drop precipitously after pregnancy. And of course, pregnancy is a stressful event. So to test that, a number of different labs, including the same lab at NIMH, took adrenalectomized rats, gave them allopregnanolone after pregnancy, and decreased a range of different behaviors which suggested that indeed allopregnanolone might be protective against postpartum depression. That was all took place in the 1980s to early 1990s. And it took then 25 years before Steve Paul finally made his way through a number of different pharmaceutical companies and got his own one and was able to test this hypothesis. And so in a landmark study, intravenous brexanolone, which is allopregnanolone, but stabilized for IV injections, could reduce postpartum depression symptoms in acute, severely depressed women who are inpatients in the hospital after pregnancy. And remarkably, this worked within a matter of 3 days, as opposed to the usual several weeks or more. And in my mind, anyway, it became one of the first true neuroscience to therapy findings. And it was approved, as I said, in 2017. So again, the reason I bring this up is because I think it's important that we take credit for the role of neuroscience in current treatments. I think psychiatry would look very, very different today if it were not for neuroscience. But we also can acknowledge at the same time that we have a long way to go. And so now I'm going to digress from direct application psychiatry and tell you a neuroscience story of the current, which is the work from my own lab. And then I'll get to how we might take work like mine and others and apply that in the future at the end of the talk. So, of course, we all know, starting about a decade and a half ago, we finally cracked the nut of genetics in psychiatry. I remember my good friends toiling away in the basement of Langley Porter. They were going to clone the bipolar gene. This is in 1992 or 3. They were going to clone the bipolar gene in 5 years in Nelson Freimer's lab. Took a long time before the field wisened up. But here is the result of that. One of the results of that, this is now already 3 years old, but of course it's a Manhattan plot of genetic risk for schizophrenia. As many of you will know, each green diamond represents a place in the genome that is statistically significantly, really beyond a shadow of a doubt, linked to schizophrenia. There are, in this graph, some 280 green diamonds. I don't know what the current number is now, somewhere up over that. Each of these, of course, confers only a very small amount of risk for schizophrenia, so it's been difficult to make progress on the biology from that. But there are a large number of other, smaller, but large number of other genes that are easier to get at the biology of. And so all those low impact risk genes are off to the right of this curve, which shows the frequency of a given risk allele in the general population on the x-axis, with the ratio, the odds ratio, or the strength of that genetic influence on the y-axis. And of course, again, those common alleles that are discovered from GWAS and on that Manhattan plot each confer a very small amount of risk. But then there's a number of different genetic factors. In yellow are single gene loss of function mutations, and in blue are copy number variants or deletions or duplications of large chunks of DNA in the genome. And these single genetic events confer comparatively massive increases of risk. And I'm going to tell you today about our work on one of those risk factors, which was also the first known single genetic event, risk, large effect size risk factor identified, the 22q11.2 microdeletion. This microdeletion is a deletion of some 25, actually 25 to 45, but there's a core region that we know gives you the full syndrome, about 25 genes on chromosome 22. It confers at least a 50-fold increase in risk for schizophrenia, although there's some debate about how strong it actually is. And it accounts probably for around 1% of schizophrenia cases. It's also, important for my own work, associated independently with working memory deficits, as well as a whole range of other phenotypes, each of which is only partially penetrant. So it's not just the neurological phenotypes that are partially penetrant in this disorder. And fortunately for us and many other scientists, most of those genes, all but two, lie next to each other on mouse chromosome 16. And through there are a number of labs, including the lab of my longtime collaborator, Joseph Gogos, that engineered a deletion of this segment of chromosome 16. And that's the model that we chose to study back when I was starting out in my own lab. And at the time, we had a still simplistic view of how to go about understanding genetic predisposition and trying to move from that genetic understanding to a full understanding of how the phenotype arrives. And at the time, I justified this by saying that we don't know where knowledge that will help us get to treatments lies within this spectrum. So we know, of course, that genes do not code for behavior. They code for proteins. And those, or in the case of 22q, there are also RNA molecules, including microRNAs that we're concerned about. Those have some action at the level of the cell. The cells group into circuits and systems, and those systems produce behavior. And so we set about in 2024, 2025 or so to try to understand this, this relationship between gene and behavior by studying across multiple levels. And because I was a systems neuroscientist, that's what we called circuit neuroscientists back then, trained in Michael's lab, I started on the behavior end and tried to work backwards. Joseph, more of a molecular biologist, started from the other end, and we met in the middle, as you'll see. We chose as our behavioral phenotype working memory because, as I said, it is disrupted in 22q syndrome, and it's also really easy to study in mice. And we chose this particular task, a T-Maze delayed non-match to sample task, where memory is encoded in a sample phase, retained for a short period of time during a delay, and then expressed during a choice phase. That enabled us separate out encoding, maintenance, and retrieval. I'm not going to talk much about that today, but we did some careful work to try to understand what the impact of the mutation and also just from a basic science perspective in the brain is doing in these three different tasks. The basic phenotype in the mice was that they took longer to learn this working memory task. So it was an acquisition deficit as opposed to an expression deficit. And it was such that mutant mice, here in green, labeled DF16 for deep deficiency on the chromosome 16, took anywhere from 2 to 3 days longer to learn the task than their wild type lettermates. Now, it had been known from much other work that this task, as well as other forms of what we call spatial working memory, rely on information exchange, cooperation, synchrony between the hippocampus and the prefrontal cortex. And so Torfi Sigurdsson, my first postdoc, showed that these mice, in the course of executing that working memory task, have deficits in the ability of the hippocampus and the prefrontal cortex to synchronize. And that's shown on this graph in one representation where he studied the local field potential, which is kind of like a Dept EEG, in both structures, and showed that across a range of frequencies, there is decreased synchrony between the two structures. And he showed, in addition, that this synchrony actually correlated with their working memory deficits, which, like most behavioral deficits in mouse models, are variable. Then Tim Spellman wanted to prove the circuit element that was responsible for that deficit in synchrony that we thought was related to the working memory deficit. So this is in the relatively early days of optogenetics, and he took an inhibitory opsin, enhanced starch 3.0, stuck it into the ventral hippocampus. Now, of course, we all know the story. He silenced the inputs into the prefrontal cortex. At that time, this is the early days of terminal silencing. So most of Tim's thesis was spent trying to prove that that neurotransmission event was actually decreased, which he did in vivo in a very elegant set of experiments, and then showed that disrupting that particular circuit element, the axons that go from the ventral part of the hippocampus to the medial prefrontal cortex, disrupted performance in the task. And that's shown here, where we have light on during different parts of the trial, and the percent correct the animals performing. These are wild type animals. I'm not yet talking about the mutants here. And you can see it has no effect in animals expressing a protein. You seeing this thing? No, you're not seeing it. Oh, well. In the gray, no effect in animals expressing an inert fluorescent protein. And when you turn on the light, either for the whole trial or during that encoding phase, you disrupt performance in the working memory task in wild type mice, but not, interesting enough, in the retrieval phase or the choice phase of the task. So this told us that information from the hippocampus to the prefrontal cortex was required for the task in order for the prefrontal cortex to encode into working memory the information it needed to be able to perform the task correctly on the subsequent retrieval phase. And Tim did a lot to show that actually it not only disrupted the behavior, but it also disrupted the representations of that memory trace within the prefrontal cortex as well. Now, that pointed to these axons that go from the hippocampus to the prefrontal cortex as an important component of the circuit that allowed working memory to take place. And I also didn't mention, it also disrupted a particular frequency range of synchrony. But that didn't link it to the mutant. That work then was done by Joseph, in fact, by a postdoc in his lab, Jun Mukai, who showed that those very axons in the mutant mice were deficient in their branches within the medial prefrontal cortex. And that, to us, linked then the circuit deficit, or the circuit element, those axons from the hippocampus and prefrontal cortex, with the inability of the two regions to synchronize and perform working memory tasks. And June even did better than that. He showed that you could rescue the branching phenotype by reversing a biochemical deficit in the tips of those axons as they grew out during development. It turns out that due to at least one and maybe two different genes that are disrupted within that microdeletion region, there's a hyperactivity of GSK3 beta, a kinase that's important for axon outgrowth and guidance in this early developmental period when the axons are forming connections within the prefrontal cortex. And so he used an antagonist and showed that he could restore normal branching if he did it early in development. So that led to an experiment to try to tie all these things together that Makoto Tamura did with June, where he treated animals with the GSK3 antagonist during the first 4 postnatal weeks at the time that these axons are developing their terminals within the prefrontal cortex, then took the drug away, waited for the animals to grow up, and implanted them with electrodes and asked, what happens to neural synchrony when you restore this axonal branching? And he showed that in mutants, there was decreased synchrony, and you rescued that synchrony with the drug. And also in mutants, it took longer to learn the task, and he rescued that behavior with the drug. He also then showed through a subsequent experiment, which I didn't think would work because I didn't think we had enough data, that it actually, that the mutants had abnormal representations within the prefrontal cortex, just like you would expect if they weren't getting appropriate hippocampal input, and that those representations were rescued as well. So this was the state of our knowledge when I left Columbia to go to NIMH. We had a hypothetical chain of events with some evidence for most of the links in the chain leading from the gene to behavior that went through axonal development into a circuit element, the hippocampal prefrontal axons, and that worked by allowing synchrony between hippocampus and the prefrontal cortex to facilitate the encoding into memory of spatial information that then led to working memory, and that fundamental disruption of axonal branching was disrupted in our mutants. There were a number of holes in that story, though. And one of the biggest holes actually goes back to that graph that I showed a while ago with multiple frequencies of synchrony that were affected. We weren't able to rescue all of the synchrony deficits, number one. Number two, that hippocampal to prefrontal axon interruption that Tim Spellman did in the lab only affected one of the frequency ranges, this one out here, gamma. And so the story that I spun just a moment ago could explain only part of the physiological phenotype and only part of the effects of rescue. And so we knew that there was more to the story. And one of the ways that we thought there might be more to the story lies in the cortical microcircuit. And so it became the focus of our attention as I moved from Columbia to NIMH. And as a postdoc that I left behind, Athir Abbas tried to suss out with his work as he finished up. So this is the canonical cortical microcircuit. It's true for most of the cortex, including the MPFC, although some of the details are better known in, say, sensory cortex than within the MPFC itself. Of course, you have VIP interneurons that inhibit somatostatin and provalbumin interneurons, both of which inhibit different parts of the pyramidal neuron. We knew from lots of other work that the hippocampus projects onto all of these neuronal subtypes, although not necessarily equally in strength. So it was possible that hippocampal to prefrontal communication and synchrony might run through any one of these different circuit elements. We had already shown that the inputs onto the pyramidal neurons were important. That was work that was done by Pia O'Neill in the lab. But we didn't know about the interneuron subtypes. So the first experiment I'm not going to tell you about, but it's published, Athir did. Again, he essentially did it when I abandoned him at Columbia, and he and two other postdocs forged ahead on their own due to the grace of my then department chair, who I have since succeeded. He studied both somatostatin and parvalbumin neurons, and he showed that the somatostatin, but not the parvalbumin positive interneurons, were important for that theta frequency synchrony and also for working memory. And that left the VIP interneurons, which we tried to study in our own lab in NIMH. And I'll point out that the somatostatin deficit, right, still allows for a role of VIP because, of course, VIP is upstream of the somatostatin neurons. VIP neurons can disinhibit pyramidal neurons by inhibiting the somatostatin neurons. So a postdoc, Shauna Silverstein, and my staff science, David Kupferschmidt, tried to tackle the problem in a really interesting way. And it was sort of trying to do two things at once, which is never a good idea. And it wasn't such a good idea in the end, but it gives us some data to look at, which are provocative. And now we're trying to just tackle one of them at once in my own lab back at Columbia. So they took an interesting all optical approach to try to understand the role of these interneurons during working memory. So in separate experiments, they used somatostatin VIP or parvalbumin pre-mice that allowed us to express a fluorescent calcium detector, GCAMP, in each of those neuronal subtypes. And then they loaded up those hippocampal inputs with a fast-acting channel rhodopsin, so they could activate the neurons, activate the hippocampal inputs, and record the responses in those neuronal subtypes, so they could measure the direct strength, if you will, of the hippocampal inputs onto each of the neuronal subtypes. And then also, because of the calcium indicator, they could image the activity of those neurons during performance of the working memory task. And indeed, all three of the neuronal types, I'm only showing two of them, somatostatin and VIP neurons in this plot, because that's the ones I'm going to focus on. But all three of these neuronal subtypes have interesting patterns of activity within the working memory task. So this plot shows you the different phases of the task. We have the encoding phase either aligned to the start or the end of the encoding phase, the delay phase, and the retrieval phase either, again, aligned to the start or the end of that retrieval phase. And you can see that somatostatin neurons in VIP in blue and VIP neurons in green, their activity varies over the course of the trial. Not too much to interpret from this. There's a couple things we could talk about, but I'll just say that this is the pattern of activity that we saw while the animals were performing the task. And I'll point out in particular one noticeable part of the activity, which is that here during the delay phase, you see a ramping up of the VIP interneuron activity and a corresponding decrease in the activity of the somatostatin neurons. Now, we also had this goal of trying to rescue the phenotype that we saw in the mutants through plasticity. So they wanted to know what would happen. Actually, Alan Park also in that interregnum period, I left him behind too. He did this really interesting plasticity experiment that I don't have time to talk about in the system. So we knew we could induce plasticity in the hippocampal inputs of the prefrontal cortex. And so, as I said, Dave and Shauna were trying to do two things at once. They were trying to understand the role of the interneurons, and they were trying to see if they could induce plasticity of the inputs onto those interneurons. And it was really hard, actually. So they ended up using a massive stimulation protocol that took 4 weeks, where they each day induced high-frequency stimulation of those hippocampal inputs. And they showed over time that you could increase the activity of somatostatin neurons in response to hippocampal input. So on the y-axis here, what you have is the test day. We did test pulses of hippocampal input and showed that the response to hippocampal input of somatostatin neurons increased over time and of VIP interneurons decreased over time as you stimulated them over the course of 4 weeks. You'll notice, by the way, we can talk about this later if you like, that even just minimal stimulation, you don't need high frequency stimulation to induce this plasticity in the somatostatin inputs. And that's something we're trying to understand. But that the VIP decrease required that high frequency stimulation. And in work that they did with Vikas Sohal here, they tried to model this circuit and understand what of the different parts of the circuit could account for the changes that they were seeing. And the conclusion that Vikas helped us draw was that a decrease in the strength of hippocampal inputs onto VIP neurons could account for the patterns of plasticity we were observing in all three interneuron subtypes. And so that led to the hypothesis that really what we're doing with this plasticity induction paradigm is affecting one of these particular synapses. And that results in changes in the efficacy of the hippocampus to drive these different circuit elements. Now, I won't show you, but it did not have statistically significant effects on working memory. So we didn't achieve our aim of altering working memory with this plasticity paradigm, although it did have a non-significant effect on that working memory that looked like an impairment. So at the same time, because again, they weren't doing multiple things at once, Dave and Shauna had tried this experiment. Sorry, I'm going too fast. The advantage of that calcium imaging protocol is you could record the activity of these interneurons not only after the animals learn the task, but throughout training as well. And remember, these neurons have this ramp of activity out through the delay period. And so we looked at all these different patterns of activity during the encoding, the delay, and the retrieval task to see whether plasticity affected any of those patterns of activity during the maze itself. Remember, the plasticity had an effect on the responses to hippocampal input, but now we're looking at the activity during the task itself. And it was all absolutely unaffected, except for one oddly specific effect early on in the training period. So early on in training, animals that had not received this stimulation had actually fall off in those VIP neurons as the end of the delay came. So this is now late, if you will, late in training. The animals learn the task, and the VIP neurons stay mostly elevated throughout the entire task, throughout the entire delay period. Early in training, that activity falls off, and only late in training does it sustain through the entire delay period. Interestingly enough, this fall off in activity was gone in the high frequency stimulation mice. So plasticity, which seemed to have a very modest, if any, effect on behavior, eliminated this drop off in VIP activity that occurred early in the delay period during learning. So Shauna shows this to me, and I say, okay, that's not terribly interesting. It's some little effect. Who knows if it's real? And how can we understand it? It was very difficult to understand. And so I wasn't particularly interested. I said, let's publish this paper, move on to your next study, and we'll do something a little simpler to try to puzzle this out. Until she showed me the data that she'd acquired from our mutant mice, which have definitive deficits in working memory behavior. And they had the same exact phenomenon. That is, this early dip in behavior, early dip in VIP activity that occurs at the end of the delay period was also gone in our DF16 mice. And in this case, I don't have it. That dip in behavior actually correlated with their acquisition deficit. That is, within the DF16s, the mice that had less of a dip took longer to learn the task, right? So that linked this dip, which I still don't understand, to the behavior. And it suggested that VIP neurons play an important role early in training, during the delay period, What could they have? Well, they're disinhibitory. Maybe they are, by dipping their activity, allowing filtering of irrelevant information during learning. So that's our hypothesis right now. We have no evidence for the hypothesis. But it was nice to see that because we, at the same time, had found other evidence that the hippocampal inputs onto VIP neurons were disrupted in our mutant mice. And this was an experiment that Dave Cooper-Schmidt had conducted in collaboration with Marissa Morales at NIDA, where they simply labeled up hippocampal inputs and asked whether there were differences between mutant mice and wild-type mice on the number of synaptic contacts onto different interneuronal subtypes within the prefrontal cortex, and showed, specific to VIP neurons, a decrease in the percentage of hippocampal terminals that made contacts onto VIP neurons compared to their wild-type littermates. So that's where we stand right now. As I told you, we have this hypothetical pathway with some evidence for each piece of it, leading through the axonal branching deficit to hippocampal prefrontal synchrony deficits into working memory deficits. And now we have some evidence to suggest that we have differential effects on different parts of that cortical microcircuit. To simplify it now, I have a new postdoc who is working on trying to actually just inhibit the VIP neurons through a very simple experiment. And we have early evidence that that may also disrupt working memory. And we don't know yet whether it's specific to the delay period or not. But that's our hypothesis, that VIP interneurons are important during that delay period for working memory acquisition and/or expression. So that's where we're at. And now I can sort of broaden out again and use these last 10 minutes or so to allow some time for questions, looking towards the future. I'm going to start, building off of that neuroscience story and talking about how we need to translate circuit neuroscience. Again, this is a place where that work is being done. And so it won't be a stretch to talk about it. And then I'll turn to computational psychiatry for a moment. So, of course, most of us think, or even thought the moment optogenetics came out as a technique, that it would be interesting to try to figure out how to directly manipulate circuits within the human brain using such technologies. And I think, the idea for that is now one that's under development, let's say. There are companies pursuing it, where you manage to find a way to specifically express effector molecules, whether it be optogenetic effector molecules or pharmacogenetic effector molecules, so that you can manipulate specific circuit elements in the human brain. You could do it invasively by injecting viruses expressing these effector molecules, or potentially even non-invasively, although that's had a setback recently with some deaths in a clinical trial of a non-invasive virus because of toxicity, I think, in the liver. And you can imagine even specific experiments. My favorite experiment in manipulating circuit neuroscience is thinking about how to reduce intractable anxiety. We know that there are fear on and fear off neurons in the central nucleus of the amygdala that have different molecular patterns of expression, in particular with regard to PKC delta. And there's evidence from primates that these same neurons govern natural variation in anxiety relevant behaviors, like is this primates? I can't remember. Yeah. Yes, this is primates. You can get a measure of anxious temperament in primates, and it correlates with PKC delta expression within the amygdala in those primates. And you can show actually that these neurons exist within the amygdala of the primates as well. And so the notion that we might be able to, say, turn on fear on neurons or turn off fear off neurons within the human amygdala is one that we could imagine doing this experiment actually today, right? So I just want to point out, and through the Brain Initiative, which of course is an important initiative the NIH has put out, we are beginning to get the tools and knowledge that we need to be able to carry out these experiments, not just in mice or primates, but also in humans as well. I will point out, though, there's another important approach that one can use besides directly using circuit level technologies in human beings that's important for us to think about, and it's the more traditional neurobiological approach for translation. So the example I'll use is going back to my own work. Let's suppose that we wanted to enhance theta frequency synchrony between the hippocampus and the prefrontal cortex in human beings to see if that would have an effect on working memory. On the one hand, we could try to induce in the hippocampus the expression of a molecule that would allow us to stimulate those inputs. On the other hand, we might also be able to learn enough about those inputs to know that, hey, there's a particular ion channel on the prefrontal neurons in the synapse that receives information from the hippocampus that's important for theta frequency synchrony. And in work actually that Nancy Padilla in the lab did, she showed that actually the hippocampal to prefrontal synapses onto pyramidal neurons filter for 8 hertz. So they're better at getting 8 hertz information at faster or slower. And so there must be something about that synapse that filters at 8 hertz. Could we enhance that with a drug to enhance theta frequency synchrony? Or could we diminish it with a drug by attacking the molecular components that allow the synapse to filter? So that's a neurobiologic approach that's based on circuits, but doesn't necessarily require us to intervene in an invasive way. And the second thought is not just molecularly, but also dynamically. So, and this is something that I know is being done here and elsewhere. If theta frequency synchrony is important, can we modulate that synchrony dynamically, either with a non-invasive technique such as TMS or an invasive technique like DBS? And I think it's important to remember that translating circuit neuroscience is not just about using circuit techniques in humans, but it's about the knowledge that we gain from circuit neuroscience and figuring out ways to translate that. And a lot of us, including my own work, we don't think enough about understanding enough about the role of those circuit elements. It's often the experiments stop at identifying them. But once we understand their role, we might learn more about the neurobiology that allows us to intervene. And then finally, which I think is the reason Matt told me to speak about when he invited me a year ago, I'll just mention that as he said in the beginning, I've been a real champion of computational approaches. These include things like biophysical modeling, which in a way, I don't know why it keeps going back, is what Vagelos helped us do on the cortical microcircuit, understanding computationally the way a neural circuit or a neuron or another part of the brain is able to represent information. Data mining, which we're all familiar with, right? And that's going to be very, very important in biomarker development. There's one other thing that we don't focus enough upon, which is computational phenotyping. And what I mean by that is constructing mathematical and theoretical models to break down behavior into its component parts. And that has an important role to play in psychiatry in a number of different ways. One of my favorite papers to talk about is Rob Rutledge's paper, where he takes mood and breaks it down to its component parts. He uses a tried and true sort of computational model of behavior, reinforcement learning, and a gambling task. And while people are playing the gambling task, they're rating their happiness. And Rob had a theory. I have a theory that happiness, thanks, there's at least one Monty Python fan in the audience. Happiness is equal to this mess on the right. It took me a while, but I finally figured out W0 is baseline happiness plus the rewards that people get while they play the task, plus the expected value. That is, what reward did they expect to get on a given trial, plus the difference between what they expected to get and what they didn't, otherwise known as reward prediction error. Those sigmas are just weighting those values for recency. So trials that just happened matter more than trials that happened 5 or 10 or 15 trials ago. And so people are playing this task, and every certain number of trials, they're rating their happiness as they play the task. And the model actually fits that data very, very well. You say, so what? And in fact, initially the result was so what, because Rob tried this in people with depression, and other than changes in baseline happiness, there were no differences between individuals with depression and people without depression in their the computational structure of their mood on these tasks. But this doesn't just tell you about happiness, right? This allows you to take happiness and break it down into these different component parts. And when you do that, and you look in particular at reward prediction error, you may not see deficits in reward prediction error per se in people with depression, but you do see differences in the way the brain represents reward prediction error. And there are activity deficits in the two main structures of the brain, the ventral striatum and the medial prefrontal cortex, that through meta-analyses have been shown to be fairly consistent in individuals with depression. And that, again, by using neurobiologic knowledge about the circuit that subserves reward prediction error, led to the prediction that kappa opioid antagonists would be good antidepressants because they might fix this reward prediction error deficit. I'd love to say that this worked, but unfortunately we just found out that a trial of kappa opioid antagonists in depression, although they did alter reward-related activity in a study that was, a multi-site study supported by NIH, that so far the kappa opioid antagonists that have been tried have failed in clinical trials. Anyway, so looking towards the future, we need more of these models, testing more mechanisms to try to get to a neuroscience-derived treatment that really does go from mechanism to clinic, maybe even more so than the brexanolone story. And that's the hope for the future from my perspective. And I'll stop there and take questions. Thank you. Sorry. These are the various incarnations of the lab. This is my lab in Columbia back in the day. This is Tim Spellman and Torpy Sigurdsson. I talked about their work. I mentioned some other folks here. This is the lab at NIH. And now this is the lab at Columbia. And everyone is, and also my collaborators on there. And I'd like to thank the NIMH for funding my work my whole career. Thanks.
Hi, thanks for this talk. This was great. So I'm speaking as someone who really struggled with being in charge of the residency neuroscience didactic curriculum for about 10 years. This is my opinion, just from having struggled with it, where I think our field does a great job, is sort of cutting edge neuroscience. Here's what's coming. This is exciting. Let's learn about new ways of studying the brain. Where I think our field really struggles, and it was difficult for me to come up with a name for it, but I would say, in my opinion, our field is, the clinical aspect of our field is not really neuroscience language ready. And I just was hoping if you could comment on that a little bit. In particular, I think DSM sometimes works against the kinds of concepts. I don't mean just doesn't involve them, I mean, works against it. And I just would be curious your comments on how our field as a whole might change our language so that a talk like yours we could bring to the next patient encounter.
So first, let me just agree with the first part of what you said. I was in your role when I was at Columbia first time around. And it was a challenge, even in our residency, which probably like yours has a number of neuroscientists in any given year, in integrating that clinically. And in fact, one of those MD PhD psychiatry residents we had almost quit in his second year because he said, psychiatry is not, like, there's no neuroscience when I go onto the wards, right? And I think it is a problem to a certain extent. I think there are ways to do it. We had at the time every quarter a grand rounds that was neuroscience in the clinic focused. It was a stretch sometimes, but I remember a particular one time there was someone who had a tumor that was impinging on the dorsal raphe, right? And they presented not with the tumor, but they presented with mood disturbance. And that was a really interesting grand rounds to think about, right? That's a slam dunk almost. But often it's a stretch, and the conversations on the wards and in the clinics are not about neuroscience. I tended to bring neuroscience into my practice when I thought patients would benefit from it in an explanatory way. And we had a lot of interest in trying to do that in discussions with the residents, but it's a real challenge and you don't really need it. You may know that there's this neuroscience-based nomenclature attempt to like change the way we talk about drugs. Instead of calling them antidepressants, call them monoaminergic drugs, et cetera. And that has some role to play in it, but you don't need neuroscience to treat patients really. And aside from neurology, and I guess some kidney docs, you don't need a lot of physiology to be a doctor in general. I guess hypertension, you kind of need it. But most of it's trial and error, just like our drugs. But I think, so getting on to the second part of your question, it really does impede advances, especially when the clinicians aren't thinking about neuroscience to be able to ask questions of their neuroscience colleagues and or begin a new study. And DSM does hurt us in that way because we tend to think of these complex syndromes as being entities in their own right that then make it difficult for us to actually think about them from a neuroscience perspective because they're complex, multifactorial, multi-system, and all the problems of DSM with heterogeneity within and across diagnoses. So I think from that perspective, a symptom-based or RDoC-like approach or computational approach to specific symptoms is more amenable to neuroscience, and we don't think about that of our patients. As you may know, right, that was one of the things I tried to champion at NIMH, was the notion that we should get more information about our patients than just our diagnoses. And we should start using things like computational phenotypes, characterize our patients that way, and see how much more information that we'll get about our patients. And again, I think a lot of practice, even if that vision were to come true, we still wouldn't have to be neuroscientists to be psychiatrists. You'd have an app that would tell us with these symptoms and this history, you might want to run these psychological tests, right? And then you hand them an iPad. And then they'd kick back probabilities and possible treatments. I mean, that's the vision anyway. You don't have to be a neuroscientist, but I think it would prompt more questions that then could be asked to explore these things if we started getting that information about our patients. And I'll just say one more thing, which is that, in psychiatry, we don't like ordering tests, right? I mean, we could be imaging every patient all the time. We could be doing functional stuff. If we were neurologists, we'd be doing that all the time, even though it wouldn't help our patients because they just want to know stuff, even when it's not going to change the practice. It's been, I don't know, Matt, you could talk about how hard it is to get, I don't know, maybe it's easy now, sequencing done for kids with autism when we know it will identify about a quarter of them, right, with a mutation. Higher?
It depends on what clinic you're in.
Depends.
10%, medical genetics, 50%.
Got it. So anyway, and there it's useful information, at least explanatory information. So we need to be more interested in doing that too, and that's a hard thing. It's hard to change culture.
I think we have time for one more question, and we can both race towards you.
Hi, Josh. Yeah, great to hear about all of your work, all in kind of a combined way. And I wonder if you have any kind of working hypothesis or idea of what the function of the neuropeptide releases by these interneurons, like vasoactive intestinal peptide and somatostatin. Maybe we must have thought about this a little bit.
Yeah. So we haven't done anything with VIP yet, but we've been doing some experiments in collaboration with Nikki Crawley at Penn on somatostatin within somatostatin neurons. And I have a grad student, Maddie Shang, who is right here, who's actually done both SST antagonists and also genetically deleting SST from SST interneurons, and really sees no effect on working memory, or actually she's also run anxiety tests and no effect of deleting somatostatin. We know it's working because it does have some robust but minor effects on locomotion in a linear back and forth maze. So we know we're doing something, but we don't seem to need the peptide for somatostatin. We haven't done anything with VIP.
Impeccable timing. It's now exactly 9:30. Thank you all. Josh, thank you so much for coming.